So we have a real treat for you for your lunch and entertainment and program. We're going to have the chance to see a fireside chat between Kyra Phillips from CNN and anchor and correspondent who you had a chance to hear from earlier, and she's going to be joined on the stage by George Scangos. And I was asked to say a few more words about George. George graduated from, got his bachelor's degree in biology from Cornell, PhD from the University of Massachusetts, and then went on to a little-known university in New Haven called Yale, where he was a postdoctoral fellow in the laboratory of Dr. Frank Ruddle. He then moved to the faculty at Johns Hopkins University, where he rose through the ranks very rapidly, becoming professor of biology at Johns Hopkins, and then moved into the for-profit world of pharmaceuticals, biotech, and spent 9 years at Bayer, ultimately holding the title of Senior Vice President of R&D and President of Bayer Biotechnology, and then he moved from there to Exelixis, where he served as President and CEO and Director for almost 14 years. Biogen was very fortunate, and Boston was very fortunate, that in June of 2010 he was recruited to Biogen as the CEO, and I think it's quite clear to everyone that Biogen is flourishing under his leadership, has important products for MS and hemophilia on the market, and we're all extremely excited about this early research finding related to Alzheimer's disease. George has also gotten involved in a number of nonprofits here in the city, the Boston Museum of Science, where he serves on the board of directors of the Biomedical Sciences Training Program, and we're also delighted at Mass General Hospital that he serves on our Research Advisory Committee. We are thrilled as a hospital at the number of collaborations we have with Biogen, and I think we recognize that it is through collaborations with companies like Biogen that we can, that our ideas have the potential to make it into the marketplace, and so we are thrilled again to be in Boston and be so proximate to companies like Biogen. George has clearly emerged as one of the most important leaders in the Boston life sciences community, and his leadership is also being felt nationally, where he was recently elected chair-elect of the Pharmaceutical Research and Manufacturers of America. And so we are delighted to welcome both Kyra and George to the podium for our luncheon program. Thank you all so much.
You know, it's interesting, George and I were just chit-chatting about our families and our lives, and we were talking about our kids and how we all want our kids to grow up to be such wonderful individuals and give back in the world. And he said, you know, sometimes I worry about my daughter maybe being a little overambitious. And I said, George, did you just hear your resume? Are you serious? But I would love to encourage all of you, if you don't mind, we will be able to take questions from the audience, and if there's any issues, I know exactly who to look to over here behind you, Chris, our main man here on Yorn. So you probably know how to do it, so if you have any questions at any time, they will pop right up here on the iPad, so please, I ask all of you to get involved in our discussion. It would be wonderful. So I'm going to put that right here to my side, and I will see when things pop up. So before we begin, one thing that I loved learning about George, when you think about his career and how amazing it has been to this point, you know, I asked him, you know, where did all this passion begin, this interest, and leading to where you are now? And I was amazed to find out that it all started at a sewage plant in graduate school. If you don't mind, George, I love this story. Yes, he rolls his eyes. He probably knew I was going to go there, but I just love this story. And it'll give you some perspective to why George is so passionate about what he's doing right now.
Okay, so this is a lesson to all of you when you talk to reporters, be careful what you say. Right?
We like the human side.
Yeah. And before I start, I do want to acknowledge the organizers of the meeting for putting the Greek flag here in the center of the top row. That's not its usual place. But anyway.
You know, the Secretary of Health and Human Services is Greek, and she's right after you.
Yeah.
So this may be a tango here.
Look, just a quick story, I won't take too long. When I was first year in graduate school, and I'd gone to graduate school kind of on the spur of the moment, and I didn't have any support my first year. And so I got a job, and my job was to go to the sewage plant every morning at rush hour for the sewage plant, dip my bucket in, bring the bucket back to a lab, purify out of that sewage bacteriophage, viruses that would grow on Pseudomonas bacteria. And these were Pseudomonas isolates that had been isolated from the lungs of cystic fibrosis kids. And cystic fibrosis kids often die from Pseudomonas infections in their lung. And so we'd get these clinical samples, plate them out. I'd find 3 or 4 different bacteriophage that would kill this particular Pseudomonas. I'd grow them up. They would get made into an aerosol and sprayed in the lungs of these kids. And lo and behold, they cleared the infections transiently for a while. The infections always came back, and they were resistant and could isolate a new set of bacteriophage that would kill them. And so this made me crazy for a year, because whatever I did, going to a movie, sleeping, going out with friends, all I could think of was there was some poor kid lying in bed somewhere with a Pseudomonas infection, having difficulty breathing, and if I were just not at the movies, maybe I could get that kid out of bed a little more quickly, right? And so I made myself crazy for a year, doing nothing but that. And so I think that was early. It was interesting for me that was a passion for me very early on, that not just doing interesting science, but doing it for some pragmatic reason in the world.
Well, I think that's the perfect segue into Alzheimer's disease and how you have been so successful in this space. And a lot of people here in the room, they heard a little bit from you about this, but they really wanted to hear more with regard to digging deeper into the trials. So if you don't mind, sort of taking us backtrack a little bit and just kind of start at the beginning, the origin of the drug development and the trials and how it led to where we are.
Okay, so you're asking me to talk about Alzheimer's disease, and I have Rudy Tanzi sitting in the front row, so, it's a little bit awkward.
Pressure.
I mean, it's a little bit awkward. So let me...
He has cue cards, by the way, if you get stuck.
So good. So correct me if I misspeak. No, look, I think, and it was mentioned this morning, I think, as part of the conference, Alzheimer's disease obviously is one of the biggest societal and health issues that we're going to have. We, as an industry, as an academic enterprise, as the whole health industry, we have to do something about this disease, right? Or it's going to be devastating to tens of millions of people and their families, and we're not going to be able to afford it, and it's a disaster waiting to happen. Been a lot of attempts over the years, and I'll talk. Our particular drug is an antibody that targets beta amyloid. There were other antibodies that came before it that either failed or had mixed results, right? Elan had an antibody, bapineuzumab, that didn't show efficacy. Lilly's solanezumab did show some encouraging data, I think, in the earlier patients in their trial, and they're repeating the trial now. And solanezumab may very well work. It may very well be the first antibody that gets onto the market. When I came to Biogen 5 years ago, there were a lot of projects going on, way too many, and we culled the pipeline quite drastically. And this project was one that clearly we kept going because we felt that the hypothesis had not been adequately tested. The hypothesis here is if you can remove beta amyloid from the plaques from the brains of Alzheimer's patients, will you have an impact on cognition or not? And in the previous trials, there had not been a clear demonstration of whether or not the antibodies did clear the plaque. And so we wanted to do a trial that would answer that question. And so we did a very careful trial where every patient was scanned, PET scanned, to make sure they had beta amyloid. We did several different doses of the antibody. We had chosen an antibody, and I had nothing to do with the choice of the antibody there. It was not long before I came to Biogen, but the antibody had some very desirable characteristics. Not all antibodies are the same. And so we were able to dose this antibody at reasonably high levels. We knew how much of it got into the central nervous system. And and we felt that the amount we were getting into the brain, at least in animal models, was sufficient to pull plaque out of the brains of animals. So we did the trial, and lo and behold, we saw a time and dose-dependent reduction in the level of plaque in the patients. I wasn't particularly surprised about that because we knew how much antibody we were getting in. We knew the antibody cleared plaque in animals, and we knew that it had characteristics. So that was encouraging data, and so we had 6-month data to show that happened. Then we had year data, and the pulling the plaque out continued. And then in a year, we were able to show that the patients on the drug did not decline cognitively as much as the control group. And that was dose dependent. The higher the dose, the less decline. And the 2 highest doses were statistically significantly different from the control. So those data are, and it was a small trial, so there may be 30 patients per group. So to actually get statistically significant data in a trial of that size means the magnitude of effect was big and the error bars were small. So it was a very nice study. AJ Verma, who's sitting here somewhere, was on a panel yesterday. Was in charge of all the imaging for that study and did a superb job. And so we were able to get clean data.
So you mentioned affordable. So a brain image is taken, and there's more amyloid than you would expect. So we know that this individual would be in the high risk range, right? Medication, $10,000 a month. You look at the numbers now, 5.5 million patients. There's going to be millions more. So how do you pay for it?
Good question. Look, if we are not thoughtful about it, it will make Sovaldi look like a blip, right? Because there are so many patients who will need this drug, and we do have to be thoughtful about it. And it can't be $10,000 a month. Let's get that off the table. You can't treat millions of people for $100,000 a year. That's just not. That's a non-starter. And we've just begun thinking about this. We've begun to have some discussions already with pharmacy benefit managers, specialty pharmacies, and other institutions to think about how we would price this so that it's affordable, so that it actually can get to the patients who need it, but it also, we're a public company. We have to take care of our shareholders. So how do we, walk that tightrope and do the right thing? And I have no answer for you yet, but I think the good news is we're thinking about it, and even before we start the phase 3 trials, right? So we'll get to an answer.
Already a number of questions coming in, and because I do have a question with regard to Alzheimer's and MS, so why don't we go ahead, and I had some notes in here about the oral MS treatment. I did get a question about what's next in your MS research.
Yeah, look, I said this yesterday, and I'll go into a little bit more detail. MS is a remarkable story. 20 years ago, and I knew people. I had a cousin 20 years ago, maybe 30 years ago, who got MS. There was nothing for that treatment, and people went downhill very quickly. There are now 10 disease-modifying therapies on the market. There are some what appear to be very promising new agents coming in clinical development, so there'll be even more. So patients and physicians have a lot of drugs from which to choose to treat MS. All of the drugs that are on the market and in late-stage clinical development slow down the immune attack on the myelin. MS is an autoimmune attack on myelin. And so for many patients, for some patients, one or more of the therapies that are on the market now actually treat them well, and they do okay. For many patients, their decline continues. It's slowed, but it continues. And they get more brain lesions, they get more cognitive impairment, they have more motion, movement disorder, and so they continue to decline. So our thought is what we don't need is more immune modulatory drugs. What we need is a drug that will actually deal with the underlying issue. And so we've been working for some years now on molecules which are intended to induce remyelination. All right? and repair the demyelinated lesions. And the thought is by combining them with a drug that does down-regulate the immune response, and now maybe shifting the equilibrium so you're net-net making new myelin, you can actually prevent patients from declining further or potentially even get them in some improvement. So the first of those molecules is a molecule called LINGO. It's an antibody targeted against a protein called LINGO. It's an anti-LINGO antibody. LINGO is an inhibitor of myelination. And the thought is by inhibiting an inhibitor, you can shift equilibrium and allow synthesis of myelin. Works great in animal models. The animal models are not a perfect representation of the human disease by any stretch of the imagination. So we got our first data in a human trial earlier this year. Those data were, I would say, encouraging. The disease is acute optic neuritis, which is an acute demyelination of the optic nerve. We treated those patients with anti-LINGO antibody. We had no impact on their vision. Their vision did not improve, so that part of the clinical trial was a failure, and I think we understand why now. But we did see improved conduction through the optic nerve and the speed of conduction through the optic nerve. And there are a number of other measures of function of the optic nerve that lead to the, I don't want to say conclusion, but let's say suggest that we did actually induce remyelination in the optic nerve. And this is giving the antibody after the insult. And it's a small trial and it's hard to infer too much, but it seems to be that the earlier the patients got this, the better the effect. So we're conducting a trial in multiple sclerosis, which is a year-long trial, where this anti-LINGO antibody is given along with Avonex, or one of our drugs to slow down immune attack. And we'll see when it's on board for a year, whether the patients who receive it fare better than the patients who don't.
So when we talk about slowing the progression, right, which is what is happening with MS and Alzheimer's, when can we start thinking about restoring function?
Well, that's what this trial is intended to do. It's intended to restore function.
And you saw glimmers of so can you give a timeline?
Well, what we can conclude in the first study is that we saw improved nerve conduction. That would be necessary for improved function. The question is whether that improved nerve conduction has a clinical impact on multiple sclerosis, and whether that improved nerve function actually is replicated in multiple sclerosis first, and if so, if it has a clinical impact or not. And the only way to do that is to ask the question and do a careful experiment and get the answer. And so we're doing it, but we'll see.
We were talking actually a little bit about wounded warriors and TBI. A question just came through since we're talking about the anti-LINGO. Could anti-LINGO have applications for TBI?
Could. It could. Look, the things like TBI or stroke they're devastating. We have a trial going on now of Tysabri, which is a very potent immune modulator in ischemic stroke. And the rationale actually would be the same as you might think about for TBI, which is that after the initial trauma, there is a post-traumatic inflammation. So there are immune cells that come in, there is inflammation, there are cytokines. These are almost certainly neurotoxic, cytotoxic events going on. And so the question is, if by down-regulating that response, if you could reduce the infarct size in stroke. And so again, the only way to answer it is to do the experiment. So we have a trial going on for Tysabri in acute ischemic stroke. And we'll get the answer this summer, actually, not very long from now.
Wow.
Now, not a phase 3 answer, it's a phase 2
Right.
study, but I think we'll get a pretty good idea of whether that works or not this summer.
Another question coming from the audience: is drug development and meaningful collaboration possible between big biotechs, pharmas, and does the government have to drive or create the platform?
Oh, does the government have to drive the platform for collaboration?
Correct.
No, I think there's more and more collaboration going on, and I think the whole economic situation now drives that. Alzheimer's disease is a big bet, right? It's expensive, and it's risky, and it's long-term. And so we have collaboration with Eisai on this, so our Alzheimer's program doesn't just include this antibody. There's a second antibody that we got from Eisai. There's a BACE inhibitor, and another mechanism from Eisai. We have inhibitors or antibodies, let's say, that recognize tau, that will be antisense molecules that recognize tau, mRNA, all of which are moving forward in development. That's a massive program. And so I think we and others are very happy to have collaborations to share the risk and obviously share the reward if it's positive. I think there's also increased incentives for industry and academic collaboration.
That's what I wanted to talk about, is when we were talking
Yeah.
about partnerships, we talked a
Yeah.
little bit about this
Yeah.
last week, and partnering with academic labs.
Yeah. Well, look, I mean, Peter said it, he put it, he reversed the way I would talk about it, that, Mass General isn't fortunate to be around the biotech industry. The reason the biotech industry is here is because Mass General and the other great institutions in Boston are here, right? And they have the base of research and the people who are the great scientists who work at those institutions. They train a lot of the people who actually work in our company now. So there's a natural affinity. And so if I look at our collaborations, they tend to be local. Right? And so, for example, we put a consortium together to work on ALS. Right? And we have a series of academic collaborations. And those are people from Harvard, Yale, Columbia, Duke, Rockefeller. And so I asked, science is an international enterprise, but those are all geographically close places. They just run down the East Coast as far as North Carolina, and that's it. And I think it's human, that people train, they tend to collaborate with people they know and trust. And so I think in the Boston area, there's a huge amount of that and a huge opportunity. We already have a lot of collaborations, and obviously I think it's to everybody's benefit to continue those and initiate additional ones.
You mentioned ALS, and you were very forthright and direct with me when we started having a conversation about failures, not just success, but failures as well. And you were very upfront about the ALS failure. Do you mind telling folks why you were not discouraged by that?
Well, we did a large phase 3 study in ALS. And we got the results, 2 years ago? Something like that. And not only did it not meet its endpoint, it was the single most negative trial I've ever seen, I think. There was no separation between the control group and the treated group.
See, I love his transparency.
They just were superimposable. And so, but this trial had generated a huge amount of excitement in the ALS community because patients are desperate. They have nothing. And this was a big trial, and we had a lot of patients clamoring to get in the trial. And it's interesting because we knew that if the trial were positive, that we would be under tremendous pressure to make that drug available to patients even before its approval. And so we spent some number of tens of $millions making drug so we would have it ready. The trial was negative. It was spectacularly negative. And I thought we owed it to patients to be upfront about it because there was so much hope and excitement that we just had to go out and say, this trial is negative. It didn't work for a small group of you. We can break out any subsets. It just doesn't work. Let's move on. So I thought we owed it to patients to be straight up about the data. It was an old-fashioned trial, right? It was not based on a genetic understanding of the disease. It was based on a drug of kind of unknown mechanism. There's some hypothesis around how the drug worked, but the science was kind of sloppy. But it had interesting data in a small phase 2 trial, which, probably we should have known better, but we didn't, obviously. And we brought it into a phase 3 trial based on the phase 2 data. It failed. That's the way drug development used to be done, right? And so, I vowed, and I think we all vowed, that was the last trial of that kind we would ever do, that we would now go back and understand mechanisms and focus on compounds where there was good science to say that intervening in that pathway would have an impact on the drug. And so in ALS, you just don't have that knowledge, right? There's some genes identified that contribute to ALS in subsets of the patients. And so we put together this consortium so we could understand more about ALS. Through the consortium, we've sequenced now the genomes of 2,000 or 3,000 ALS patients.
You've invested a lot in gene therapy in the past year.
Well, this is sequencing their genome, and we have invested in gene therapy as well. But we've identified a couple new genes involved in ALS. And I think we have our hands on a couple of potentially interesting targets against which we're now trying to make drugs to see if they'll
Such as?
have
Could
an
you give
impact.
some examples?
No.
I'm trying.
Good try. but
Moving
anyway,
on to audience questions. Here we go. The story about medical need is expensive. How do we address similar need for ultra-rare genetic disorders, which therapies are attainable, but that they're commercially attractive?
Yeah, look, I mean, I don't know how to take that from an issue of drug pricing or technically what's feasible. Most of the ultra-orphan genetic diseases, or many of them, are monogenic. Right? I think all those monogenic therapies will eventually be open to gene therapy. Right? And especially with gene editing technologies, have been, I think, discussed here at some point. I think over the next few years we'll see treatments for many of those. You're also seeing little companies like Spark in Philadelphia that's treating congenital blindness. They have some really interesting data there. Very dramatic videos. I don't know if you've seen these videos of kids who have one eye treated, and so they can see out of one eye, the treated eye, and not the other, and they walk through mazes. It's dramatic, the change it can make in people's lives. If there are 5 patients in the world, then it's not a disease that Pfizer should take on, right? It just, from an economic reason. On the other hand, certainly people in academia, and I think most of the people in our company have as their first thought what we can do for patients. And that's why we're here. When we were in a conference room and people came in, it's a very dramatic moment. You get in a conference room and you've spent hundreds of $millions and some years on a phase 3 trial. It's blinded. And there comes a moment in time where the statisticians come in, they know the answer, and they tell you the answer. Right? And if it's positive, you break out the champagne. But in the case of the ALS trial, it was negative. There were tears. There were tears in the room.
People broke down.
People broke down in that room. And I guarantee you, nobody was thinking about how much less money we were going to make. People were thinking about the hope that they'd had to be able to do something for those patients, and that hope was now dashed. And so I think that we as an industry have a responsibility to do a number of things. Do what we can for diseases where we have some technical chance of working. Look, the other thing I think we have a responsibility for is to make sure, to do what we can to make sure the patients who take our drugs actually benefit from them. Right? Drugs get tested in a clinical trial, and you get something, it reduced, the rate of decline by 30%. So the FDA approves that drug. But that doesn't mean every patient had a 30% improvement. Some patients might just stop declining at all. Other patients probably get no benefit at all. And the problem is in some diseases it's easy and rapid to distinguish that. In other cases it's not. But over time we need to figure that out, right? Because patients who are on a drug that doesn't benefit them still get the side effects. They don't then get delayed from getting on a drug that actually does help them. And so I know that's all precision medicine, a lot of discussion, but I think there's a moral imperative there to do that.
We're going to talk more about precision medicine, by the way, with the Secretary of Health and Human Services coming up actually after our discussion, so we can go more. Maybe you stick around and throw some questions out,
Okay.
George. I'd appreciate it. It looks like we have time for one more question.
Wow.
And why don't we go ahead and look ahead, let's say let's give it 5 years. Your biggest concern about Biogen's business?
In 5 years? No, look, my biggest concern actually doesn't stem from Biogen. It stems from the environment in which Biogen operates, because we, Biogen, and the industry and the academic enterprise are at an amazing point in history. I believe that 100 years from now, when people look back, this is going to be a time of amazing advancement and transformation. And it's coming at a time when we have increasing economic pressure on the whole enterprise. NIH funding is not even close to being adequate. There's increased pressure on drug prices everywhere. So everybody involved in the creation of new therapies is under economic pressure at the time when there's amazing opportunities to treat all kinds of diseases. So we need thoughtful, intelligent public policy, right, in order for both the academic enterprise and industry to be in a situation where they actually can fulfill their potential. And I worry that we're not going to get that.
I think that's a perfect segue to the Secretary of Health and Human Services coming up after our discussion. George, thank you so much. Really appreciate it. And thank you we had so many questions, I couldn't get them all in. Thank you very, very much. George, thank you.
Thanks.